Okay, so we're going to start with this chapter 13. But before we start, I want to kind of tell you what we're doing here. Chapter 13, 14, 15, and 16. The core idea here is we're looking at the nuts and the bolts of genetics. Last test, we talked about more of a big picture idea of how genetics worked. Now we're doing specific molecular level discussions about how genetics works. So that kind of frames this entire test. Okay? So we kind of start this with what? A little bit of a history lesson, right? What came before this? Well, we talked about Mendel, right? We said that Mendel talked about heredity and there's something moving around. Okay. This chapter and this section talks about how we discovered two very important things. First of all, that DNA is the molecule of heredity. Not RNA, not protein, but DNA is the molecule of heredity. That's the chromosome theory of inheritance from the 1900s to the 1940s in there. Showed that DNA was the molecule of heredity. The second important point, or the second important thing here is what? How DNA is a molecule of heredity. Starting in the 1950s and 60s, we found that what? DNA, we call it the central dogma of molecular biology. DNA codes for RNA, RNA codes for protein. And it uses that triplet codon. Okay? So those are two very important things that we found. Now let's kind of start this off with, Messner, 1800s, found DNA in the nuclei. Good. First important thing here is what? 1900s, Griffith's experiments. So what Griffith did was what? He showed that there was some type of molecule of heredity. He didn't know it was DNA, didn't know it was RNA, didn't know it was protein, didn't know anything about it. He just was able to show that there is a molecule of heredity that was moving around. So how did he do that? You kind of need to know this experiment. Okay? So, starting with panel A, rough bacteria. He put this rough bacteria into a mouse, and the mouse lived. Okay. The rough bacteria here, it's the exterior shell of it. When you get into phagocytosis, that means that it can kind of get around the bacteria. Okay, that's another chapter, another section. But basically, he put up one bacterium in, and the mouse lived. That's panel A. He had another bacterium, very similar to that rough one, but this was smooth. And because of its smooth shape, it could evade the immune system of the mouse, and it would kill the mouse. Okay, so he put the smooth bacterium into the mouse. Mouse died. That's panel B. Okay, rough bacteria lives, smooth bacteria, the mouse dies. Got that. So what he did was next. He took that smooth bacterium, that bad bacteria, and he chopped it up and broke all that bacteria apart and put it in the mouse. Well, he killed the bacteria. So guess what? The bacteria did not kill the mouse because he killed all the bacteria. It was bad bacteria, right? But since he killed it, he broke it apart, it didn't hurt the mouse. Okay, panel A, B, and C, no problem. Everything expected. Panel D is where it gets kind of interesting. So what he did was he put the rough bacteria, right, that he in panel A is fine. That rough bacteria, non-pathogenic, not harmful. But then he mixed it with the remnants of the smooth bacterium. Okay? So when he put rough bacteria in the mouse lived. When he put broken apart smooth bacterium in the mouse lived. But what if he put the rough bacteria and the broken apart smooth bacteria together and then put that into the mouse? Well, you'd think if neither of them were harmful, that would not be harmful either. But it was harmful. Okay? It was harmful, and that's the critical point here. That rough bacteria somehow could take up, absorb, what we call it, be transformed, some of what was in that smooth bacterium and take on the characteristic of the smooth bacterium. Okay? There's some type of molecule of heredity that was in the smooth bacterium that could transmit to the rough bacterium and make that rough bacterium into a pathogen. Okay? The important point here is what? That last one. The smooth cells were releasing something that could change, we call transform, the non-harmful rough bacterium into the harmful smooth bacterium. Now, Griffith didn't know what that was. Okay. Unfortunately, Griffith died during the Blitz, so he couldn't continue the experiments. But something was there. Something is transmitting. Now, we know that today to be DNA, but at the time, this was not known. Okay? But something was transmitting between the smooth bacterium and the rough bacterium. Okay? So what does Griffith show us? That there's some molecule of heredity that can transmit between organisms, in this case, bacteria. Okay. So in the 1940s, Avery and Riccardi, they continue his work. Okay? So what did they do? Well, they said there's something moving around, right? We know there's something. And we know the four macromolecules are lipids, proteins, RNA, and DNA. Okay? So we know there's four macromolecules. So let's selectively eliminate each one of those and see what happens. So they destroy the lipids, the fats. They repeat this experiment right here, and they get the same result. So they know that that molecule of heredity is not a lipid. Okay? They do the same thing with protein. They destroy all the protein, but leave the lipids, RNA, and DNA. Experiment number 2, same thing happened. That rough became smooth. By destroying the protein, it didn't affect the experiment that implied that protein's not the molecule of heredity. They repeated another time with RNA. Okay, they destroy all the RNA. No change. That implies that RNA is not the molecule of heredity. So then they do it with DNA. Okay, they destroyed the DNA but left the RNA, protein, and lipids. And what do they see? Bingo. That little experiment I just showed you did not work that way. That implied that DNA was the molecule of heredity. That DNA contained the information of genetics. And we know that to be true today. Okay. Key point here is by process of elimination, they said that DNA was the transforming particle, or the transforming principle. Okay. They didn't know the mechanism. Okay. They didn't have an idea of RNA and protein and DNA. They didn't have any kind of idea. This just said that, hey, DNA is a molecule of heredity. Okay. Another set of experiments in the 50s. These experiments used labeled atoms. Okay? Labeled atoms. One labeling was sulfur. Sulfur is found in protein. Okay? Another labeling was phosphorus. That's found in DNA and RNA. Okay? They use this bacteriophage, and it's a virus that affects a bacterium. Okay? And they traced the tag. Okay? When they traced the tag on protein, they saw that that protein tag did not go inside the bacterium, implying that it was not the molecule of heredity. But the DNA tag did, again implying that the molecule of heredity was, in fact, DNA. Okay. So that's what the Hershey and Chase did, is they essentially confirmed that DNA was likely the molecule of heredity. Okay. So we get to the 1950s. Lots of scientists in the United States, England, France, trying to get that how does it all work? Okay, had an idea, maybe it was some people thought it was protein. Okay, Some people thought it was DNA. A lot of people thought it was DNA. Some people thought it was protein. Wasn't sure exactly what was going on. And there was this race to find how, genetics works on the molecular level. Okay, and that's what this is talking about. Pauling was one. Watson and Crick, obviously, they were some. Rosalind Franklin was another one. We talked about how Watson and Crick did this and how Watson went around other labs and looked in other people's, learned some things here, learned some things there. Watson and Crick put it all together with some other people, and they found the structure of DNA. Okay. So, what's the structure? Well, I should say this. They not only did they find the structure of DNA, they found two things. One, it was a double helix. Okay, some people like Pauling thought it was a triple helix. Okay, but they found it was a double helix in an anti-parallel configuration. And if you read the original paper, they kind of hint at what we call the central dogma of molecular biology with RNA and protein and the codons. Okay. So they found the nucleotide, and they also found how these things are structured. Okay, so let's talk about nucleotides. You need to know the definition of a nucleotide. Know what a nucleotide is. Okay? Not only do you need to know a nucleotide, you need to know all four bases: A, T, G, and C. Adenine, guanine, thymine, and cytosine. You also need to know the base pairing rules. A matches with T, G matches with C. You have to know those complementary rules. You also have to know what a purine is and what a pyrimidine is. A purine, okay, let's look at what the purines are. See the picture on the right? And we see 3 P's and an orange pentagon, and it looks like almost like a little balloon and 2 blue circles, pentagon type things. Okay, those blue looks like balloons hanging on a string. Those are the bases. Purines, okay, purines are the 2 on the bottoms. Purines have 1 ring. Pyrimidines, I'm sorry. Purines are on the top. Purines have 2 rings. Pyrimidines, thymine and cytosine, those have 1 ring. Okay. So what's the take home here? Know what a purine is. A purine has 2 rings, and it's the A and the G. Know what a pyrimidine is. Pyrimidines have 1 ring. They are the thymine and cytosine. In RNA, that thymine is going to be called uracil. Okay. So go here. Okay, so Chargraph's rule. Remember what we said? You have to know the base pairing rules. A matches with T, G matches with C. You have to know Chargraph's rules. Chargraph said that the amount of A equals the amount of T, and the amount of G equals the amount of C. And that kind of makes sense because those are the ones that base pair, right? So, if you have 30% thymine in your DNA sequence, you're going to have 30% adenosine. Okay? Because A matches with T, so everywhere there's a T, there's an A. What's left over will be the amount of GC in the guanine cytosine. So I worked out an example here. Okay, if you have 30% adenine, what is the percentage of the rest of the bases? And you can expect to see a question like this. Well, Chargraph said the amount of A equals the amount of T. If there's 30% adenine in the DNA, then there has to be 30% thymine. You know, if the amount of A equals the amount of T, well, if there's 30% A, then there has to be 30% T. So if we add all those two up, we get 60% AT. So what does that leave left over? Well, 40% has to be GC. And if we know the amount of G equals the amount of C, then we can say that there's 20% G and 20% C. And these GCATs are, of course, the bases of DNA. Okay. Rosalind Franklin, we kind of hit on her before, kind of finish it up. Her contribution was X-ray crystallography. She showed that it was probably a helix. Okay, this was a very critical piece of information for Watson and later Crick to understand the molecule of DNA. There is some debate that she should be in the Nobel Prize that they won. There's also a little bit of debate that they looked at her research notebook without her permission and took her information without her permission. And she passed away at 37, maybe even very likely because of her experiments. She was working with X-rays, and, well, she got cancer. So, anyhow, just know that there is some debate that she very well may be deserving of a Nobel Prize. That they do not award Nobel Prizes posthumously, so it's a moot point. Okay. So this is the molecule of DNA. Okay. What is DNA? DNA is two chains of nucleotides. Okay, we call them strands. And they're running anti-parallel, or we say in opposite directions. Okay. Look at that picture. See it has DNA on that picture on the right? Okay. Do you see how on the left of that picture, at the left corner of the picture to the right, there's an orange pentagon. And at the very top of that, there's an O. At the bottom, there's a little HO. Okay? Now, look on the other side. Look at the bottom right corner. Do you see how the little O is at the bottom of that? But then on the left, the O is at the top? Okay, that is showing the anti-parallel nature of DNA, or the opposite direction. That these strands have direction. And you see how these strands, one strand has the O at the top, and then the other strand has the O at the bottom? That is just showing that DNA is running anti-parallel. So what's the take-home here? 1, DNA is made of 2 strands of nucleotides. That's the first take-home. 2, those 2 strands are anti-parallel to each other, meaning one's going up, one's going down, and that's shown in the picture right there. Number 3, those 2 strands are held together with hydrogen bonds. You have to know that too. The 2 strands are held together with hydrogen bonds. Okay? 3 points you got to know. Okay. We talked about this before. Is the base pairing rules. A pairs with T. G pairs with C in DNA. Okay. So let's look at this example. We have DNA at the top. It says GACT. See that? Well, the opposing strand would be CTGA. Okay, you see how there's a GACT and then CTGA? One's at the top, one's at the bottom. Well, let's go back to this picture. That's really saying that the strand on the left and the strand on the right at this picture on your right bottom is the strand at the top and the strand at the bottom here. That's what it's trying to show you. Okay. What's the take-home? The take-home is this. In DNA, A pairs with T and G pairs with C. That's the take-home point. Expect to see a question that says something like, a DNA molecule is GGGCCTTA. what is its complement? Okay, or what's the opposing strand? And you have to know the base pairing rules. You have to know the base pairing rules. I'll say that again. You have to know the base pairing rules. A pairs with T, G pairs with C. You might even see a diagram like this, where I might take that, let's just say that middle picture, ATCG, okay? I might have that picture and then the bottom one absent. And then I might say, what would pair with this? And you'd have to say TAGC. Something like that. Okay. So, DNA replication. What do we know about DNA replication? Well, we're going to talk about the molecular nature of DNA replication. We talked about this with mitosis and meiosis, or meiosis, that, DNA replication has to happen. Okay, so we're going to talk about the specifics. Specifically, we have two strands of DNA, right? Going back to this picture, we have two strands. See how there's one on the top, one on the bottom? In this picture, there's one on the left, one on the right. So DNA has two strands of nucleotides. Okay. Each of those two strands, when DNA replicates, will serve as the template. It will serve as what the free nucleotides will build on. Okay, I got a picture here in a second, but take home is that each of the strands of DNA, when it's replicated, is template for the new strands. And again, I think that's going to make more sense in the next picture. Okay. Next point here is one template builds continuously and the other one discontinuously in segments. Okay. And third, each new DNA molecule consists of one old strand and one new strand. Now, I've got a picture coming up here, and this is going to make a lot more sense when we see that picture. Okay. Okay, so let's look at this picture. Okay, see this picture, and it says lagging strand, leading strand. Okay. What did we say first? The first point was what? Each strand of the DNA double helix is template. So that first point, let's look on the right of this picture. You see where it says topoisomerase and the little twistiness to it? Okay. What that topoisomerase is doing is it's splitting apart that DNA strand. And as the DNA strand splits apart, it makes a fork or a replication fork. And each of that single strand that comes off from that blue arrow and then past that will serve as the template, okay, as the original base sequence of the new sequence added. Okay. Point 2. One template builds discontinuously, the other one continuously. Okay, see how it says lagging strand and leading strand? The leading strand is always being made. Okay, that polymerase is always making that. The lagging strand on the top, the DNA polymerase has to go from left to right. It has to continuously make short stretches, stop, loop back. Make a short stretch, stop, loop back. It's discontinuous. Okay, what's the take home? Take home is this. One template builds continuously, the other one discontinuously. Okay. And then the third point is kind of goes with the first point. That, see how it says leading strand and lagging strand at the far left? If you could trace back the origins of that double helix at the top and the bottom, half of that double helix would be the DNA on the right. Half of that double helix is the DNA is new DNA. Okay. We call that semi-conservative replication. Another take-home point. Semi-conservative replication. Half of the DNA is new, half of the DNA is old. Okay. Some important molecules here with replication. These are just a few of them. There's actually several more. these are the important ones. DNA helicase. We said that these strands of nucleotides are held together with hydrogen bonds. The helicase, which is that blue arrow, that's essentially going to pull apart those hydrogen bonds and expose the bases. DNA polymerase. DNA polymerase is going to add free nucleotides. Okay, the nucleotides that will then pair with the exposed template and make a new strand of nucleotides. And then third, DNA ligase. That's going to be like glue. That's going to glue together our DNA and make it a contiguous strand. Okay, so just know these three enzymes. So this is basically what we just talked about, how these strands come apart and come together. Okay, so we got what? 2 strands, and this is complementary. G matches with C, T matches with A, right? We pull those apart, and what happens? Well, we pull these apart. Okay, let's start over here. Okay. We have our old strand. We have 2 strands, one on the left, one on the right. Helicase pulls these apart. Okay, it breaks those hydrogen bonds in the middle. Okay. DNA polymerase is going to add these nucleotides in. Okay, we have a leading strand and a lagging strand. The free nucleotides come in based on the old strand or the light blue strand. Okay, they match with the base pairing rules: G to C and T to A. Then finally, that ligase is going to come in and glue those together, seal them up, and make the light purple into a DNA strand unto itself. And you notice that new DNA strand is complementary to the old strand. It follows the same base pairing rules. Another picture of that idea of semi-conservative replication. Okay, the dark blue on the top is the old DNA. That DNA is going to split the replication fork, and what comes out of that replication fork is half new and half old. That is semi-conservative replication. Semi-conservative replication is that DNA that is replicated is half new and half old. Okay. Discontinuous and continuous. We kind of talked about this before, but I just want to, hit it one more time. Synthesis of new DNA is happening on both strands of DNA. Okay? But here's the difference. On one strand, it's always happening. You see on that, it looks like a zipper, and on the left, that 3', that 3, that replication is continuous. Notice on the right, though, see how it's going, instead of going up, it's pointing down? It's having to make short stretches and then loop back and make another short stretch. That's discontinuous. Okay, so just know what continuous and discontinuous are. Checking for mistakes. Now, see how we see these bases adding here and these bases adding here? And let's go back to this picture right here. See how these bases come in? G and C and T and A, and it looks like it works. Well, guess what, guys? Errors happen, mistakes happen. And sometimes a mistake can happen, and instead of a T and an A coming together, you might have a T and a T, or a T and a G. Okay? So that's where error correction comes in. The DNA polymerase can scan the DNA that's just been made, and it will say, figuratively speaking, of course, you're not a right base pair. If you're not correct, I'm going to pull out that incorrect one and put in the correct one. If it was not for this mechanism, we would not have, we would have mutations. We'd have too many mutations. Now, does an error get through occasionally? Yeah, sure does. But not enough to be a detriment to life. It allows enough mutations to have, evolution and act upon, but it's not too much. Okay. And then that's well, I should have mentioned this before, but this is just the finishing up with Franklin, and we talked about that. Okay, and that concludes chapter 13.